So in this final video on phylogenetics, we're going to complete our discussion on the idea of interpreting relationships. So we'll entitle this last flowchart, Interpreting Relationships, and of course we mean evolutionary relationships, Roman numeral 2, and we'll complete on some final ideas about evolutionary relationships and phylogeny altogether. So, final idea that we have to understand is the following. When we're inferring phylogeny, when we're trying to, sort of guess how evolutionary history plays itself out through this study called phylogeny, when inferring phylogeny, most of the time we're going to be using derived characteristics. And if we do that, if we use these derived characteristics, we have to understand a couple of key points. So when we're inferring phylogeny, evolutionary history, through those derived characteristics, we need to understand that the shared derived characteristics, SDC, are all going to be unique to the specific clade in question. And that clade is that group of organisms all from that same common ancestor. Oftentimes, utilizing derived characteristics means that there are going to be outgroups on this phylogenetic tree that you're utilizing and trying to make. And an outgroup is simply a species that is exactly how the name sounds, that diverged. So it's an outgroup, it's not within the group. Species that diverged before the lineage in question, or before lineage being studied. So let's say you're studying a certain lineage, a certain class of organisms, a group of organisms. There's going to always be an outgroup or a couple of outgroups that are just not relevant to your study, and we call them outgroups for that reason. And then, of course, there are also going to be, if you're looking at derived characteristics, those that are, of course, following the derived characteristics in question. There are going to be in-groups, and those are going to include the species that you are studying, species being studied. So those are the things that you care about. And finally, in order to create a phylogeny, in order to create a tree based off of derived characteristics, you utilize something known as a character table. So this is a quantification method, a way to quantify shared derived characteristics. Specifically, you're going to complete a table of sorts that's necessary in order to compare. And it's there in order to compare the characteristics of different species. You use this table to compare characteristics of different species. What do I mean by this? Well, let's say you have species 1 and species 2, and you notice that a certain character between them is absent. The fact that they do not share a certain character, you give that score, that table, that part of the table, a 0. An absent character means that there's a 0 going to be shown up on the table that you're creating. And then if the character is present, this is a binary. It's very simple. If the character is present in the comparison that you're looking at, you would give the score of 1. Your textbook has a good figure of this character table, and once you've created enough characters and you have looked at, enough characters and compared enough characters with this binary code, you can then finally make a phylogenetic tree. Much easier said than done. I've simplified the process greatly. It takes a lot of time, a lot of effort to do this, but nonetheless, it's a great way to look at phylogenetics as a whole, utilizing derived characteristics. In addition, when we're interpreting relationships, oftentimes we look at tree length in the sense that we look at the branches specifically. What do I mean by this? More specifically, when studying these relationships, oftentimes you will encounter trees with what we call proportional branch lengths. So we'll write proportional branch lengths. There is a reason why sometimes the lengths of the branches on these phylogenetic trees are proportional in nature. Most of the time what you're going to notice is that some trees are going to have the branch length, and that branch length will be absolutely irrelevant. It will be not equal, sometimes at least, to the degree of evolution. Okay, so this is a scenario in which we do not see a branch length being proportional, and we have to be sure to say that, yes, this does happen sometimes. Not proportional, not equal to the degree of evolutionary change And that evolutionary change is specifically in regards to each lineage in question. So that's the sort of side note that we have to have. Not all are proportional, and a lot of the times if they're not proportional, that means the branch length has no influence and no equality or equity to the degree of evolutionary change. Okay, now that we've got that out of the way, now we can actually look at the ones that are proportional. So we'll say sometimes we do see this proportionality, and this proportionality is going to be in reference to something. Well, that's of course in reference to evolutionary change. Sometimes this branch length that we see on the tree is proportional to evo change, or it's even proportional sometimes to even time itself. And when we see that, we can easily see through those branches a reflection of the number of genetic changes that these diverging, let's say, parts of the tree have undergone. So it's a good way to see the number of genetic changes. The longer the branch, let's say, the more genetic changes that are going on. And we can also state that furthermore, from this idea of different number of genetic changes, we can state that different lengths of these proportional branches will indicate that we have different evolutionary rates. And if we have different evolutionary rates, i.e. if we have a short branch, we have a fast evolutionary rate, quick evolution. If we have a long branch, we have a long evolutionary rate. This is going to all be seen in different lineages. So we'll just write that down over here, in different lineages. Don't get hung up on this idea of understanding the idea of proportionality and how it relates specifically to evolutionary change. You don't need to know the details. Just understand that sometimes proportionality does show up, and if it does show up, proportional branches will reflect genetic changes, and different lengths will mean different evolutionary rates, all within different lineages of organisms. Finally, sometimes the branch itself can not only represent evolutionary change, but sometimes it can even represent time itself. So sometimes the branch is equal to time in some trees. And if it's equal to time in some trees, we can actually use fossil data. Everybody's favorite, fossil data, nice and accurate data. Use fossil data for branch points. For those nodes are not going to be based off of divergence of organisms, but those nodes, because branches are equal to time, are going to be based off of points at which we have different time scales, different time events. Use fossil data for branch points, and we know that those branch points, since we've built a tree before, are the nodes, in other words. Finally, the last point that we're going to talk about, and it's a good way to really summarize this whole lecture, is the fact that you've seen a lot of things that might not seem relevant to you. I understand that completely. You've seen a lot of facts and figures and ideas, but what we have to understand is the scope of what we're learning. The scope of what we're learning is simple. The fact of the matter is that there is lots of data around us. There's lots of data from all of the organisms that we see, from the fossil records that we see, from all of this idea of evolution, presents us with lots of data, and lots of data is going to be combined with the idea that we see lots and lots, let me rewrite that plus sign over here, lots and lots of species. So lots of species. And so if we see all of this information that's thrown out at us, sometimes what happens is we get really complicated comparisons. We start seeing very, very complicated comparisons. Things get difficult. We get lost in all the data. You might have gotten lost in all of this phylogeny talk. How do we make sense of it? Today we have the ability to take all of this that we've learned today, all of this phylogenetic ideas and themes, and use computer analysis to make sense of it. Now, that does not mean that what we've learned is useless because the computer knows it. What it means is that the computer can take the amount of data that we have, the amount of species that we know about, and analyze it at a much faster rate than we can possibly do. The computer will spit out things that give us two basic ideas. Computer analysis to create a phylogenetic tree, in essence, when we want a phylogenetic tree, can be devoted to two main components. Either you can want maximum, what we call parsimony. So the computer analysis, based off of this data and the species that it gets, can either give you max parsimony or max likelihood. And both of these maximums are in regards to a phylogenetic tree. Do you want a phylogenetic tree with maximum parsimony, or do you want a phylogenetic tree with maximum likelihood? Let's see what each of these means in a little bit more detail. Maximum parsimony is simple, and that is a very easy way to understand it, because it simply means, and I'm using the word simple for a reason, is that parsimony is the idea that simple is better. And we can get a little philosophical here. You can even state that maximum parsimony is based off of a philosophical theory that we call Occam's razor. So I don't want to get any too deep in the philosophy of this, but this simply means that the less assumptions that we make, the fewer assumptions, the fewer complications that we have, the better the tree will look. The better the tree will actually represent life, because life itself just happens. And the evolutionary relationships that we see just happen. So keep everything rather simple, keep everything parsimonious is what we can say. In this situation, we simply want the most, what we call parsimonious tree. And that simply means we want the simplest tree, most parsimonious, let me make sure I spell that right, tree. And if we have the most parsimonious tree, we have the tree, and I can't write it here, but you can write down with the fewest assumptions, and thus the fewest changes, and thus the simplest tree there. So we don't want to get hung up on this philosophy. Just understand that if you want a phylogenetic tree, you can either want the simplest, or you can want the one that is the most likely. Both of these have their own positives and benefits depending on what type of phylogeneticist you're talking to. We don't need to get into those details. Finally, maximum likelihood. This type of phylogenetic tree is based on the exact probability rules. It's based on probability rules, so it's not based on simplicity, but it's based on probability rules on how DNA, so it's the DNA, we have very good DNA sequencing techniques. We're going to utilize them, combine them with the computer analysis, combine them with all this data of species that we have, and we base the probability rules on how DNA sequences change over time. And if we know how they change over time, we can input it into a computer. The computer can tell us how we expect it, how likely we see the changes over time, and thus how likely we see the phylogenetic tree that results. So overall, I know there's a lot of, it seems relatively irrelevant information, but trust me, it's not. All of these tools that we're utilizing are a nice harmonious conclusion to the idea of evolution itself. All the way through Darwinian, micro and macro evolution can be summarized and utilized through the study of phylogenetics.